Okay, I got 3 or 4 minutes after, so we're going to go ahead and get started. My name is Brian Behringer. I'm from Christian Brothers University. I'm the Director of Entrepreneurship and Innovation at Christian Brothers University, and I'm very happy to be helping out Ryan and Erica from Bio10 and the Scipreneur, Memphis Scipreneur Challenge to bring you some entrepreneurial training related to real life stuff. So we're going to be talking about intellectual property that comes from our IP partners. And we're going to give an introduction and kind of explain how everything works. But this should be a very intense next 9 weeks. So if you're looking for a challenge, this is definitely going to be up your alley. And you're going to be working on real world stuff and learning real world skills from an entrepreneurial perspective. So this is, less about the science and more about the business. But that's why we're all here, to try to learn how to launch this technology into the marketplace for our respective technologies and entities. So we're really looking forward to it. We're going to dig right into it here right now. So we'll jump over here to this session right here. All right, so one thing just to know in Zoom, if you go to the top right-hand corner and hit the view button, it's where it is on mine. You can change that view to a speaker view, and then my screen will get a little bit larger, which will help you to follow along. Right now I'm seeing, everyone's faces, but this way we can do it with seeing my screen. Okay, so welcome to the Memphis Scipreneur Challenge. We're going to get started here with just basically some introductions, and I'll hand it over to Ryan to kick that off.
All right, thanks, Brian. First, I want to thank everyone for being here this evening. Hopefully everyone is staying safe and warm, and hopefully trying to enjoy the snow a little bit. I also want to thank all of our sponsors, because I know without them, none of this would be possible. From our standpoint, I think it's a pretty good thing we're virtual right now. Otherwise, I'm really not sure how today would have happened. So for anyone who wasn't here last week, I'm going to go ahead and give it a very abridged intro about myself, the program, and sort of what our expectations are. So for those who don't know me, my name is Ryan Hughes, and I'm this year's director of Memphis Scipreneur. I was a part of the organizational team last year and was a participant 2 years ago. And so with this being the 5th time we've held this event in Memphis, I've basically been involved with the program for more than half of its existence. And as such, I've seen from both a participant's and an organizer's point of view just how valuable this program can be, especially for those from a science background who have aspirations of getting into entrepreneurship, or just want to learn more about entrepreneurship, or other career path opportunities. And I think putting the program together this year, it has been a challenge. I think life has been a challenge for most everyone. And I know early on there were questions of whether or not Scipreneur would be able to happen this year. But we've really been able to bring on an amazing team, and I'm really thankful for the help and the support we've received. So even though this year's program will be entirely virtual, I actually believe we've put together our most comprehensive and actionable program to date. So I'm really excited for everyone to participate, and I think you guys are going to learn a lot and have a ton of fun. So real briefly, before I turn it over to Erica, I just want to emphasize that this environment is completely casual. If we were meeting in person, we'd have catered food and adult beverages. So please don't feel like you can't do the same during these meetings. Feel free to eat and drink. If you have to work in lab, continue to do so. Wear your pajamas, I don't care. Just whatever you feel comfortable doing, because we want you here. We want you to relax. But most importantly, we want you to learn and have fun. But with that being said, we do expect you to actively participate week in and week out. There are a lot of people, the faculty members, the faculty mentors who are present today included, who have donated their time, to be a part of this program. So please respect that by giving us your attention every single week. And, remember, at the end, there's a competition where the top 3 teams win money. And with each person on that team could win up to $500. So in my experience, the teams that win are the ones who put in the time and really apply what they learned during the course of this program. So again, thank you for being here. We're all looking forward to a fun and exciting 9 weeks. So with that, I'll turn it over to Erica.
Thanks, Ryan. Good evening, everyone. My name is Erica Dillard. I am the current president of Biotin Academic Alliance Memphis Chapter. There are 2 other chapters in the state, one in Knoxville and one in Nashville. We all have Scipreneur challenges in our respective chapters, but Memphis is the one that's been going strong for the longest, and this is our 5th year. So really, really excited about moving into a new phase. Pandemic has really changed the way that we do a lot of things, obviously, and including this hasn't been immune to the effects of the pandemic. But I think with those challenges, we've really put together a program that I think you'll find to be really awesome. One of the main points that I've been stressing in terms of our chapter here in Memphis is trying to encourage collaboration and partnerships with the other institutions in the city. We have a very strong relationship with St. Jude and UT and University of Memphis even, but some of those relationships we really have been trying to mature with the other institutions in the city, and this is just a manifestation of that exact thing, working with Brian, who is the director of the Center of Entrepreneurship and Innovation over at CBU, really have a strong curriculum for you guys that I think you're going to absolutely love. I wish this was something that I had as a grad student. I also came through UT as a medical student and a graduate student. Dr. Pfeffer, who was on my committee for my thesis, so it's awesome to see him here. So I'm definitely a product of Memphis, and really, really am excited about having something like this here that you guys can take a part of. So with that being said, I'm not going to linger too long, but just want to welcome everyone, give you a warm welcome, and just let you know that, again, just to reiterate what Ryan said, this is laid back. This is time for you to really digest and learn some of those things that you would build upon if you are looking to really go into entrepreneurship and maybe even try your hand in industry. Biotin is an organization, it's a nonprofit that's focused on increasing the workforce here in the state in biotechnology, STEM careers. And the Academic Alliance is a way that we do that by providing outreach to our graduate students and our postdocs here in the city. And this is one of our staple activities, and I think this is one of the main ways that we're able to provide that service to our membership. So thank you again for being here, and I am going to pitch it over to, is it Shelby or maybe Brian? I don't know. So thank you.
Go ahead, Shelby. Introduce yourself.
Hey, my name is Shelby Pranich. I'm the Associate Director at the Center for Entrepreneurship and Innovation at CBU, so I'm Brian's right-hand person. I have been involved with pitch practices, pitch review, some of the sessions for Scipreneur Challenge in the past, mostly in the past 3 years. So I'm really excited to take more of an active role and be assisting Brian, Erica, Ryan, all of the people that we have scheduled to slot in for office hours and for sessions this year. You can reach out to me at any time, any questions you have. I'll try and get those answered as best as I can for you. Yeah, I'm just really excited to be here. I think that the participation that we have this year definitely surpasses what my expectations were for this being virtual. I've run countless programs for entrepreneurs that are in person, so I'm really happy to see the turnout for tonight. So yeah, I'm here. If you guys have any questions, you'll see my face a lot, and we'll be in contact a lot over the next 9 weeks. So thank you for being here. I'm excited to see what all of you get out of this.
Thanks, Shelby. Okay. So now you see a big screen of me. My name is Brian Behringer. I am a serial entrepreneur, 3rd generation. I don't think I had a choice in the matter because my dad put me to work at around 12 or 13. But I've never looked back on my career with any kind of regret, even with the successes have been good enough, but the failures have been painful enough to teach me some very valuable lessons, which I actually cherish probably in some ways more than the successes for what I do now, which is as the Director of Entrepreneurship and Innovation at Christian Brothers University and formerly University of Memphis. I've been able to transition into the academic world. I even got the professor sweater and everything. to teach young adults, those in business, people like yourselves, anybody that is willing to listen on how to be an entrepreneur. And it's been a rewarding transition for me because I get to see and visit and work with teams and create something and watch them create something. And that's really what we're going to go through this entire 9-week process is an exercise in creativity and an understanding of how to take that creativity. And that is something that actually is a skill that can be learned. So even if you don't think you're creative, we're going to show you ways of doing that. So back to what I do. This is what I do. I teach entrepreneurship. We build curriculum for CBU as well as online and for other universities related to the entrepreneurship and innovation. We launch businesses. We work with programs like Bio10 and the 800 Initiative. We work with the city and the county, all to kind of profess and encourage the prosperity of entrepreneurs and helping them along the way. So we hope you enjoy the journey and what you learn here. We do think it's very practical skills. And without further ado, we're going to go ahead and get started. But before we do, I got 2 things to say. One is we're going to go to the next slide. I'm going to hand it back over to Ryan. He's going to introduce our partners and sponsors. And then we also, one other thing is if you look in the chat, there's an Icebreaker link. We're going to actually do 2 or 3, or we're going to try these out. There's going to be the first time Shelby and I have done a virtual Icebreaker. But there's a Google Doc link in there in the chat. Click on it and answer the question. We're going to collect the information and talk about it a little bit later. We're going to do that 2 more times. One time we'll ask you to just kind of think of some things. The other one we'll send out here right after we get some feedback from this current forum. So Shelby, when you're ready to send out the next forum, just go ahead and do that in the next 5 or 6 minutes or so after we get some in for this one. All right, so going back to the presentation, I'm going to now hand it back over to Ryan. He's going to introduce our sponsors and other participants in our program.
Thanks, Brian. So our main sponsor is Bio10 Academic Alliance. They've really been critical in making sure that this program gets going this year and that we have all the resources we need in order to make this program work, especially virtually. And then all of our intellectual property partners at St. Jude Children's Research Hospital, University of Tennessee Health Science Center, and the University of Memphis. And then again, shout out to Brian and Shelby at CBU. I mean, I don't think without them this would be going as smoothly as it has been. And I think they've built a really robust and amazing platform for this year's curriculum. So I can't thank them enough, and we're really excited to have them on board.
Well, thanks, Ryan. You set the stage and the bar pretty high, so I'm not sure this is going to be a completely flawless situation, but that's one of the biggest and best things about being an entrepreneur is just kind of rolling with the punches, and that's how we'll progress along. Okay, so tonight is all about understanding what intellectual property we're going to be working on through these next 9 weeks. I'll also provide some links at the end for our website, the Scipreneur website, that has a full schedule that discusses everything that we're going to go through week to week. We're going to have 8 weeks starting this week where we discuss different topics around entrepreneurship as it relates to what you're trying to build from a company's perspective, as well as your technology. We'll also work towards a tail end on how to build a narrative and pitch your idea in a fashion that investors and customers and business partners that you have to try to convince to do business with you will, receive well, right? They will understand by the way you pitch the idea and your overall grasp of entrepreneurship and the different facets that make up a viable solution. That's all we're going to be talking about over the next 7-8 weeks, and then we'll go into a presentation format, kind of a Shark Tank type thing, with a panel of judges at the end. And as Ryan said, there's monetary prizes on the line. So, I mean, if you're doing this just for the money, hey, here we go. That's a great way of doing it. We know that you're also going to benefit from a lot of good entrepreneurial education and a lot of exposure to different technologies, a lot of networking with other individuals that are in the same kind of position you're in, wanting to try this out, wanting to build a solution. We even have a participant or two that are not from these institutions that could plug into this, that have a wealth of other experience. If you weren't there for the Q&A session last week, we did discuss that we're going to have guest speakers. and other subject matter experts come on during certain sessions during this process. They're going to talk about different things related to regulatory and kind of the federal outlook on how to get a life sciences type technology to the market. We're going to have people talk about funding and financing, those two separate documents or topics. One is about how to finance your company and how to build a sustainable company as it relates to the financial side of things. And then also we'll talk about funding. We'll have a venture capitalist here in town come on and talk to us about funding and how venture capitalists think. And that's a big part of understanding how to win success and gain money, is to understand how your customer thinks. And we're going to talk about that early on as well as in the funding process. We'll also have office hours every Friday. If you can see down at the bottom of your screen right now, we're going to have office hours tomorrow from 3 to 5. We'll send out a link to sign up for those. There'll be sessions you sign up for. And I'll be available during that time period to answer any questions. It might not be the best thing for y'all to do tomorrow just because you haven't gotten really started yet. You might want to utilize that time to meet with your team and start working, but we will have office hours every Friday. And when we have those subject matter experts talking on the Thursday night, they're actually going to join us the next day for office hours as well. So if you didn't get an opportunity to talk to them that night before, you will have an opportunity to talk to them the next day. And then also we'll give out contact information because after all, this is about building your network. And so knowing some of the individuals that we're going to put in front of you could actually have long-lasting benefit to your careers as entrepreneurs if that's what you so choose to do, and we hope you do. So without further ado, and Shelby, if you can do me a favor, I've got the main screen up so I can see what I'm doing. If you see anybody raising their hands or if anybody is raising their little virtual hand in Zoom, if you can send me a chat, just let me know there's a question. That would make it easier. Another way to do it is to come off mute, but let's try this muted for the thing and then throwing a message over to Shelby about your question, and then she'll throw the question at me and then I'll see it, and then we can answer it that way. We'll have a more open dialogue at the end. But if you have a really pressing question, everything else, go ahead and throw it in chat, raise your hand, do whatever you need to do, get our attention, and we'll jump to it real quick. So this is one of my favorite quotes about entrepreneurship. It's also, kind of one of those things where if you think this is dangerous and really crazy to even think about, then entrepreneurship it may not be for you. And obviously we're not literally jumping out of planes, but an entrepreneur is someone who is willing to jump off a cliff or out of a plane and build a plane on the way down, right? That's kind of what we do. We have ideas, and generally they're half-baked, and half-baked is being generous. And then we push forward and we figure out how to build out the solution incremental step by step. Each problem, coming up with a solution, working that solution into some sort of process related to the growth or protection of the business. And that's what entrepreneurs do. We continuously evolve our solutions because we set expectations for our customers, and those expectations change with our customers the more they use our products or our services because they're used to that norm and they want more and more and more. So as an entrepreneur, you're always working towards improving your product and keeping up to pace with what your customer needs. And we're going to talk about customer development and customer discovery and how important that is during this process. So this is kind of a fun quote, but it does, if you kind of envision what we're talking about here, it is very similar from a metaphorical perspective of how entrepreneurs think and the willingness that we have to kind of take a leap of faith. And I will say as a side note, 10 years ago, taking a leap of faith, giving up a paycheck, giving up an opportunity to have a long-lasting career with a major business, that was probably not the best advice that people could give entrepreneurs. These days, with the volatility of our market, the volatility of our economy, our state of affairs as a nation, our state of affairs as it relates to as a globe, as a planet, is ripe for opportunities to either voluntarily or involuntarily become an entrepreneur. So if you were to ever be faced with some sort of significant roadblock in your life where a career path didn't work out, entrepreneurship and starting a business is a viable solution for that and can take just very little money to get started. So it's always a viable option to become an entrepreneur. Also, having these skills inside of an organization. I myself worked for FedEx 3 different times over 12 years. I worked at St. Jude for a few years in their IT department building solutions. These were always times where I went back because I wanted that paycheck, but I was always thrust in a position of entrepreneurship at those positions. So having the skills that I've had and Shelby has and a lot of us that do, it's allowed us to be very portable and transferable from thing to thing, because the way we think doesn't change, only the problems that we solve are the things that change. I like this quote or this definition of entrepreneurship much better, and I think it will be a little bit more explanatory of what we're doing. Entrepreneurship is an attempt to create value through recognition of business opportunity, the management of risk-taking appropriate to the opportunity, and through the communicative and management skills to mobilize human, financial, material resources necessary to bring a project to fruition. Notice it says project. You could take out the word project, you could add product, you could add solution, you could add a vaccine, you could add anything you want, but all the things leading up to that word are what we're talking about from an entrepreneurship perspective. It's mustering up the resources that you need, whether you have them innately in your own self or in your own bank account, or for 99.99% of the rest of the world, where you have to go convince other people to not only buy something from you, but potentially join your team as another founder or as an employee. You have to convince funding resources, whether it's institutional or a bank, to give you money. And then you have to convince customers to buy that solution. So it's all about mustering and pulling together all of the resources you need to create to take an opportunity to market. And I think that's a big thing. And the word create is important to us in this regard as well, because we talked about that a few seconds ago about how to be creative. We all know that solving problems, and you're all in the same boat in this regard. You're scientists and people that are working on technologies. You're going to face problems with your technologies and your intellectual property, and you go about right now fixing those and adding to those, looking at the data, trying to come up with new ways of delivering the solution to a level of success and efficacy in your trials. That's the same thing we're talking about now. We're just talking about it from a business perspective. So I've never met anybody that couldn't teach how to be creative. I've never met anybody who I didn't think could be an entrepreneur, whether they were willing to or not, but at least understand it. And I never, unfortunately, I've always had the opportunity and the success of teaching individuals where I felt like they walked away with skills that would help them regardless of the path they want to take, whether that's owning their own business, working in an early stage startup, working in a small business, or going to a Fortune 50 company or a pharmaceutical company and working inside of that environment. So these skills are transferable and transportable in large and wide ways. Okay, so here's the week agenda for today. It's also on the right-hand side. We're going to do an icebreaker here pretty soon. Shelby, you give me the high sign when you're ready, if you have the data, and then we're going to go through the IP parade real quickly. And when I say real quickly, we're going to go in depth. We're going to spend about 45 minutes on the whole thing. We'll go through each one of them. You can submit questions as we go. As Ryan said, he's going to try to table the questions to just a few for each piece of the intellectual property, just for sake of time. We are going to take 2 hours tonight to do all this, but it'll be about 45, 50 minutes that we go over the IP parade. You're going to have another Google form that we'll send you that you're going to be able to choose which intellectual property you would like to work with. So there'll be 6 to choose from. You'll pick from 1 to 6, and then we'll do our magic and put you into teams, taking your desire into account. Is everybody going to get their choice number 1? Odds are not in that favor, unless we just have everyone differently choose number 1s. So we'll give you your first and second. Maybe some of you might get third, but that's the way we're going to do it. Shelby and I've done this several times. Usually everyone is fairly decently happy with the choices that they made and the choices that we made to put you on the teams. We'll go into another icebreaker, a treasure hunt of sorts. We'll send out more information in the chat about that as well. Number 5, we'll do the team reveal. So while we're doing the icebreaker and a few other things, maybe answering some questions, we'll come back with the teams. We'll do one more icebreaker before it's all said and done, and then we'll end up with a Q&A session and also allow y'all to break out into teams so I can build breakout rooms in Zoom, put you in there, and then you can kind of get to know each other, and I'll leave the Zoom session open as long as I need to, and Shelby and I will pop in to those breakout rooms just to see if you have any questions for at least 20 or 30 minutes after the session is over tonight. Okay, so I don't see any questions coming through chat, so we'll just move on to Icebreaker One. So in the form you were sent, you have a Zoom challenge. We've been living in this virtual state for the last 12 months, a little over a year now. And, not only has Zoom and all these other companies' finances gone through the roof, our level of experience with what goes well and what goes poorly inside of a Zoom session have greatly increased as well. I know I've seen some rather funny things, just including last week where the lawyer logged into his meeting with the judge, and his secretary or friend or daughter, I forget who it was, put a cat filter on his laptop. And so during the whole session with the judge, he kept saying, "I'm not a cat. I'm not a cat." I thought that was pretty darn funny, because it's just him and the judge with a couple of screens, and he literally looks like a cat, and his mouth is moving and everything else. So it was kind of funny. So that's what this first icebreaker is. We're going to read a few of those out. Shelby, if you're ready.
I am ready. So, of course, humor is objective, subjective, whatever, but I chose a few of these that are the funniest out of all of your responses. And I've been sitting here for the last 15 minutes laughing to myself about some of these and getting embarrassed because I can't believe that some of y'all have seen this stuff on Zoom. But, so I have a lot of people said the cat lawyer thing, which was hilarious. So since Brian already read that one, of course that's one. Another one that was really funny is someone said a professor screenshot his picture of him listening to a lecture and used it as his Zoom profile picture, so it looked like he was always intrigued. I love that. That's really good. Another one was a woman who, where's this one? The lady that started gossiping with her friend on the phone about her friend that was a nymphomaniac and her detailed experiences, assuming that her Zoom mic and speaker was on mute. I would die if that happened to me. And then the third one was another person who thought that their camera was off, which makes me so paranoid to always double check if your camera's off or not. I saw a video on social media of a lady who used the restroom during a work Zoom meeting and she thought her camera was off. You guys have some amazing stories. Those are my top three, though. I thought those were the funniest out of all of them.
Yeah, those are great. So many of those ones we've seen with kids doing the dancing, the mob type dancing on their classrooms and driving their kids crazy. People accidentally standing up, not realizing they only had a button down shirt on and no pants on. Kids coming into the room. I was on a conference call yesterday with Rory from the Small Business Center here, and his daughter joined us for at least 50% of the session. And as the session grew, she realized that no one was paying attention to her, so her volume just kept going up and up and up until we all said, "Oh yeah," and said something to her. So it was quite funny. But I also think that all these Zoom sessions have actually allowed us to connect in ways we haven't before. So I love the funny things. We'll keep doing these kind of icebreakers through the sessions just to break things up. I appreciate everyone's submission to that regard, and a brief chuckle about all the things that happened. And of course, the words spoken the most in the last 12 months is "your mute button's on," right? Or "I'm sorry, I was on mute." I mean, I think those were probably, I've said those, well, let's not count how many times I've said those. Okay, so moving on, what we're going to do now is I'm going to hand it over to Ryan, and Ryan's going to walk through the intellectual property that we're talking about. Ryan, do you need me to share or assign you as host so you can share anything, or are you just going to talk through it?
No, I'm just going to talk, and I think the way we're going to do it is every presenter is just going to share their screen, and so each of them will have their own individual presentation. So I don't know if you need to make them host for each one who goes.
Yeah, just you. How about this? You tell me which one's going when. I will make that person the host as soon as you tell me, and then they can share.
Okay, I'm in no particular order. Looks like Dr. Pfeffer, you're the first one on my screen.
Oh, thank you so much.
Okay.
I'm going to do this very informally because I didn't put together a presentation.
Okay.
But I thought I would just explain the project. And I'm interested in glioblastoma, which is a form of brain cancer. It's the most devastating type of brain cancer. And it's a kind of a personal thing for me because I had a very close friend when I was working in New York who was at Cornell University who died of glioblastoma. I noticed there was something strange in some of his behavior in the lab, and unfortunately was diagnosed with glioblastoma, and within 6 months he died. And so the I've been working on this form of brain cancer for the past 20 years, and Erica was doing some stuff with me a while back when she was a medical student. And so this IP derives from a project that was derives from one of my graduate students' thesis projects. We're interested in this part of the brain cancer cell population that's called glioblastoma stem cells. They don't form an individual, but they have stem-like characteristics. And so when we were trying to figure out what's dysregulated in glioblastoma, we identified this protein called BRG1, which is Brahma-related gene 1. And it turns out that it has two important functional domains, a bromodomain and an ATPase domain. It's part of the mammalian SY-SNF complex. Don't want to get into this that much, but it turns out that there was a great interest in this protein a while back. Pfizer made a drug called PFI3. PFI is for Pfizer inhibitor, and they gave up on it because they tested it in brain cancer. It didn't kill the brain cancer cells, didn't kill any kind of cancer, and they dropped the project. Well, we were looking at glioblastoma stem cells. We found out that if you treat the cells, if we knock out BRG1 in brain cancer cells and treat them with the standard therapy in glioblastoma, temozolomide, they died. They were really dramatically sensitized to it. Then we took glioblastoma stem cells, knocked out BRG1, and they were also very sensitive. So then we said, okay, let's try to see if we can treat one of these domains with an inhibitor. So we tried the old Pfizer drug, which they dropped, and it turns out PFI3 worked really well in sensitizing these cells to temozolomide. So our IP is, well, you can't do anything with the Pfizer drug because even though they never got a patent, but they own the IP on it. But so we made changes with a colleague at UT who's a chemical biologist. He made a variety of PFI3 structures. We tested those, and we have patented a series of these compounds, and all of these sensitize glioblastoma in tissue culture and also in animal models. So if we inject glioblastoma stem cells into the brains of mice and treat them with this PFI3-like substance, and then treat them with temozolomide, we prolong the life of the mice dramatically. And to just give you a feel of how bad things are in glioblastoma, the major change in treatment of patients with glioblastoma is this tumor-treating field, which prolongs people's lives by less than 3 months. So it's a pretty crappy disease. So our drug, when we do animal studies, prolongs the life by over 50%. So that's a big deal. And we think that this is an interesting set of substances. We're hoping that we can find a partner, a venture capitalist, or some company that'd be willing to support our research so we can move this from the preclinical stage to the clinical stage. And it turns out this whole day today I was on a Zoom call, and just to give you a feel about how bad glioblastoma is, the NCI has a separate whole set of grants for GBM where they're trying to find new therapeutics to improve the prognosis for glioblastoma patients. So I think this would be a very important drug for people, as well as profitable for some biotech company if they decide to take up this substance, new set of substances. I hope that was clear enough.
Yeah, no, that was perfect. Thank you, Dr. Pepper. So we're going to go ahead and open it up for questions, and we're going to limit it to 2 questions per speaker. You can choose to either type it in the chat box and I can ask the question, or you can go ahead and ask the question yourself. And if you can't think of the question now, you think of something later, feel free to go ahead and type something in and we can get it to him. Looks like, okay, that's not a question. That's just a statement in the chat box. I guess I can start with one. How different structurally is your compound versus PFI3?
We made modifications in one part of the ring structure of this that would increase its stability, and so it's reasonably different that it is separate intellect. You know, we are, the patent is pending on the drugs that we made, so the patent attorneys have said that it is different enough that it is patentable, and those drugs have dramatically better efficacy than PFI3.
Great. Thank you.
I'm not a chemist, so bad at this.
Any other questions? Okay, again, if you think of them, just go ahead and type them in the chat box at any time during tonight's program. Again, thank you so much, and we'll go ahead and move on. Let's see. Looks like Dr. Zachary, you're next up on my list.
Okay, well, I see if I can share my screen here.
Yeah, let me, Dr. Zachary, let me make you the host, so you'll have the share things as well. It should be coming your way right now.
Okay, yes, I see it.
Perfect.
Okay. So I do have a little slideshow, and I'll just start by telling you a quick background. I work as a pediatric occupational therapist. I'm also the chair of the OT department at UT Health Science Center. And when I was practicing as an OT in the 90s, it was the mid to late 90s, we noticed, other therapists and I, there was a very large increase in children who had fine motor skill delays, postural weakness. Some of them even had like poor head and neck control, and there was an increase in children who had diagnosis of cranial asymmetry, which was flat spots on the head. And we really weren't sure what was going on, and were really curious. So when I did my dissertation, that was the topic of my dissertation. So that's kind of the background of how I became interested in this topic. And now I'm going to tell you about the tummy time trainer that I developed. So if you Google "help, my baby hates tummy time," or if you Google tummy time, you're going to see "help, my baby hates tummy time." And it's very common. The reason is before 1992, babies slept on their stomachs typically. But around that time, the American Academy of Pediatrics discovered that infants who had died of SIDS were more likely to be on their stomachs than their backs. So they recommended that babies sleep on their backs moving forward, and that did drastically reduce SIDS. But then they, as well as we therapists, started noticing some issues, and it was not necessarily from the back sleeping, but what was happening was when a baby sleeps on their stomach and they wake up in the crib, they naturally push up on their hands. They look around, they use their neck control, their muscles in their arms, their weight bearing, so they're getting stronger, as opposed to when they're on their back and they wake up, they're looking straight up, so they don't use any of those muscles. So they typically, the parent will try to put them in tummy time when they're awake, and they cry because they're weak. They can't hold their head up. They don't have this muscle strength to hold their head up and move around. So there, since that AAP recommendation, even though yes, there's been a great reduction with SIDS. There has been a 60% increase in infants diagnosed with cranial asymmetry, which is flat spots on the head. And that is from not enough tummy time in some cases. In some cases, it's before they're born, the way they're positioned in the womb. But in many cases, if they don't get enough time on their stomachs and they spend too much time on their backs or in equipment, baby gear, then they end up with these flat spots on their head and have to wear a helmet, like you can see in the middle photo. And then what we saw as therapists, we started seeing motor skill delays. So kids who had just this low tone, kind of weak upper extremities, sitting with their poor posture. This was in the late 90s, so the AAP had recommended back to sleep in 92. So that was about the time these kids hit school, and I was working as the school system therapist. We'd also see children who couldn't hold their pencil correctly. And it turns out that our theory and my theory is that it's because they don't get that weight bearing on their hands from when they're younger during tummy time. And many of them even don't end up crawling. They just go straight to walking. So the tummy time definitely promotes motor skill development, but the problem is many babies just don't tolerate it. For my dissertation, I did some research, survey research, and found that up to 60% of babies cry when they're placed in tummy time. They just don't tolerate it. And no parent wants to hear their baby cry. So it's usually they pick them up and then they just don't worry about it. So the other tummy time systems that are out there, you can see these images. You have the big hoop, kind of like the hula hoop top one, and then they also have this prop pillow that's a C-shaped prop pillow, but it can put pressure under the infant's arms, which has, sensitive nerves, lymph nodes, and blood vessels. It doesn't even look comfortable. If you look at the images of the babies on there, it seems a little awkward. And the one thing that I don't like about the arched top is that just encourages the baby to roll over on their back so they can see the toys. So it's not encouraging tummy time. So I started when I was working with infants who didn't like tummy time, I started thinking about some other options, and I came up with the tummy time trainer. And so this is, it's made out of foam, and I just basically cut out the foam to have two little spots for the arms so there wouldn't be the pressure under the arms. And then it's molded, you can't really see it, but it's molded to the infant's body so that it kind of is a soft position, but it shifts their weight toward their bottom, which helps them to get their head up and into a better head position, and then they end up liking it better. And then there's the play gym that's in front that's just a triangle foam wedge that has the mirror and toys. And so I made one of these for my niece, and she loved it and tolerated tummy time much better using it. And then I tried it with some of my clients, and it was successful. So I brought it to UT, and UT Research Foundation submitted a patent application for the tummy time trainer in December of 2020. And the major markets are parents. New parents spend an average of $7,000 to $9,000 on baby equipment a year. A lot of it is unnecessary and is not developmentally appropriate or stimulating. So, but this would be something that would be good for development. Also, child care centers and pediatric therapy centers, early intervention service providers could use this. And as far as product licensing potential, Graco, Fisher-Price, Baby Einstein, Bright Starts, Infantino, Kidco are some companies that might be interested, hopefully. And then just a little about me. I came to UT in 2013, and I have since then had two books published by the American Academy of Pediatrics, Retro Baby and Retro Toddler. And the real focus of the Retro Baby book is tummy time and cutting back on baby gear to promote development. There's an entire chapter on tummy time and how important it is. So that's a little bit about me. I have a website and have quite a few followers on social media, and so that's been helpful with the book, getting the books promoted. So I'm open for questions now.
All right, thank you, Dr. Zachary.
Oh, I will add, I'm working on a pilot study to have 25 families with an infant use the tummy time system and provide feedback. I'm working on getting it through the IRB right now.
What ages do you recommend for tummy time? Like what age do you no longer need to do it?
So parents should start tummy time from birth when they get home, but that when they're really young and have a low birth weight can be done on the parent's chest. So if the parents lies back, reclines, and then puts the baby on their chest so they're face to face. Then when the baby gets to be around 10 pounds, you can start putting, you could put them on the floor before that, but they're probably not going to have enough head control and they're going to fuss. So around 10 pounds, you could put them on the floor on one of these, tummy time trainers, and then they usually are until they roll over. So 5 to 6 months, they need to be getting that time, and once they start rolling over and crawling, then they're building on their development.
Okay, and we have one question in the chat. How much tummy time is required for an infant?
The AAP recommends 30 minutes a day, but that doesn't have to happen all at one time. It happens in small increments, so I recommend to parents to do it after a diaper change or maybe like right after a bath, and then build up to longer periods of time. So minimum is really 30 minutes a day. In my survey research, the average infant got less than 15 minutes per day, though, unfortunately.
Okay, thank you so much. If there's no more questions, we'll go ahead and move on to, let's see who's going to win this time. Dr. Huang from the University of Memphis.
Hello. Hi. Okay.
Dr. Wong, do you need me to share let you share the screen, or do you have anything to present?
Yes.
Okay,
I have some slides.
I'm gonna make you co-host, right?
You.
Now you should see it.
Yeah.
Okay, great.
Okay. Let me present. Okay, hold on. Can you see? Sorry, can you still see? I don't see my slides. Okay. No, I can see.
We could see your PowerPoint application. We couldn't see the presentation if you turned it into presentation mode. Or we saw something, but it looked all zoomed in. So if you just want to show us in your PowerPoint app and just kind of cursor through it, we can do that too. Yeah.
So, can you see now?
Yeah, we can see it really well.
Okay. Yes.
There you go. We see that too.
Yeah. Okay, thank you. Okay, I really appreciate this opportunity to introduce my technology. First, I want to thank Ryan to contact me, and I'm very glad to be one of the mentors. So today, our technology is called Surface Enhanced Raman Scattering Single Vesicle Technology. We call SERS SVT. So this is a technology that profile exosomes in biological fluid. So let me give you a little bit of background. What are exosomes? So exosomes are membrane-bound extracellular vesicles that are constantly released by almost all type of cells. So the unique thing about exosomes is they carry the molecular contents such as proteins and nucleic acid from the parent cells. So that means exosomes are a very promising biomarker for disease diagnostics and monitor for example, cancer. So that's also it's the clinical significance is emerging as one of the most promising analytes to develop next generation liquid biopsy for cancer diagnostic and monitoring. Especially it addresses one of the major challenges in cancer is early detection, because exosomes are constantly released from the parent cells. So when cancer cells develop, they constantly release and dump into the blood. So if we draw the blood and analyze exosome in the plasma, so we can catch the exosome from tumor cell in the background of a band of normal exosomes. So this is a very promising and new analyze for new generation diagnostic and disease monitoring without tissue biopsy. We know that tissue biopsies have some limitations. For example, after remove the tumor, you don't have anything to look at. So, but however, we can constantly monitor the tumor, the patient recovery and even the tumor recurrence through the blood test with the exosomes. So, however, even despite the significance in the clinical applications, to detect exosome is not easy because they are very small, like a 200 nanometer, less than 200 nanometer in diameter. Mostly it's like 30 to 150 nanometer. So traditionally, the optical microscope is very hard to see. So we cannot analyze like tissue staining and cell use a regular optical microscope. Most important is that the major challenge is that the tumor-derived exosomes in the body fluid, they're mixed with abundant of the exosome from normal cells. So if you use the bark method, you cannot see these exosomes from the tumor. So they are masked by the band of the healthy exosomes. So bark method is very limited to analyze the exosome for disease, purpose. So what we are trying to do is develop a simple method. So for example, here are the exosomes from plasma. So you can see that we have small portion from the tumor. So we have to analyze each individually, so we can catch up with small portion from the tumor. So, and also this not only for the clinical application, but also for cell biology, to understand the tumor biology, and also some other fundamental questions we have not addressed in the cell biology and the tumor biology. So this is a very good the way to further help to address, to advance the field of cancer. So, and what our technology here is we call SIRS SVT. So it uses a very small palm size the chip. We coat it with a gold film, and then we separate use a template into multiple wells. So here is the mechanism of the detection. So to show in each well, so we functionize the gold surface with antibodies that only catch the exosomes. We know that in the plasma, there are a bunch of stuff, the proteins, cell fragments, DNA, RNA, all those kind of things. However, we only catch exosome because the exosome, they have a molecular marker on the surface. One of the marker is CD81. So we use antibodies, so we can specifically capture exosome directly from plasma without the pre-purification. So after that, so we target the So we analyze, because we want to find out the tumor exosomes. When you see a tumor marker, protein marker on the surface, we know that most protein marker, they're on the cell membrane. So we just try to use antibodies, so primer antibody to recognize whatever surface marker we want to detect. And then after that, we use our gold ladder probe. This gold ladder probe linked with the secondary antibody. And also, we call it a source probe. So whatever primer antibody is not here is different, and then this is universal. This gold ladder probe linked with secondary antibody. From the only gold ladder part, we coated with a Raman reporter. So this is optical probe. So once we hit laser, and this will give Raman signals. Raman is an optical signal, generated from this reporter. We encode on the surface of nanoparticles. This Raman signal is very, very sensitive. So it allows the detection of a single particle. So that is, we can detect a single protein on the surface of these small exosomes. So we developed a method because we want to quantify the fraction of exon from tumor. So that means how many percentage of exon from tumor. So we need a mask. So we use the dark field. The exosome, they scatter under light microscope, very high resolution microscope. So we know that where is the exosome. And then we use laser, Raman laser, when we detect the Raman signals. So exosome capture universal exon, the disease exon and the healthy exon, all of them is here. But only those from the disease exosome with targeted biomarker, and they show up in the Raman imaging. So we use the image. So you can see here, you see less compared to the total exome, because some of the exome, they don't have the cancer marker. So by this dual imaging method, we analyze, we can quantify, we profile the amount of exosome with a specific target marker. For example, shown here is a HER2, is a breast cancer marker. So we can profile, and this profile, we can quantify the fraction of the subtype exosomes, FT. So it's like 1/3 or just 10% or 5% come from tumor. So another one is like a mean expression protein level, like the expression of this marker on the single exosome cross all the total exosome we analyze. So this information is, especially for this one, is very good parameter for the not only for the diagnostic monitor, but also for basic, tumor biology studies. So I'm going to quickly say we built up, this microscope. This is a general microscope, but we use laser, Raman laser to excite, and that's why we can see the exosome, both dark field and Raman imaging. So this dual imaging gives that's the key feature. So dual imaging to detect, profile exosomes. So the key feature of our technology. So what we do is to try to quantify surface protein on single exosomes. Further to quantify the amount of disease exosome in the plasma. And also we use plasma directly. So we only use like 8 micrometer per test from plasma directly. So without purification. This is also portable because very small chip. So each patient or multiple patients, just one is usable, we use it and throw away. So it's a portable device. And high throughput is like ELISA. So on a single chip, we can analyze multiple samples simultaneously. And also quick, from the once we put plasma capture exome to detection, only 5 hours, turnaround time. And like I said, it's a huge market. All the clinical labs and researchers and this cancer center doctors, everybody can benefit, can use this. for their own purpose. So I want to show you one example. We use our technology is to detect the HER2 subtype of breast cancer. We know that HER2 is one of the major subtype in breast cancer. So this is a patient data. So we use like a 20 patient data and also with healthy donor 10. And you can see here's why is our technology is a fraction of the HER2. The fraction of HER2 positive exosome in healthy, you can see is about 20%. We know that HER2 is a growth marker, so they also show on the healthy cells. However, when we come to the early stage, the stage 1 and 2 patient, we see this HER2 expression among the exosome increases significantly, almost to like 50%. And at the stage 3, and they slightly increase more. So from here, p-value, we know that our technology can detect breast cancer, the HER2 subtype, at the early stage. So that means stage 1 and 2. And this is another one is a protein expression level. From here, you can see that using the protein expression level, this is not good Glioblastoma marker. The p-value here from early stage to healthy is more than 0.005. So that means we are not able to, if we use this HER2 protein level. But however, if we use a fraction of the HER2 expression exosome, we can detect the early stage. And of course, those parameters we can detect at stage 3. So that's a locally advanced stage. Stage 3, you can see, compared to the healthy, p-value is smaller than 0.005. And we also compare with the standard ELISA. ELISA is a protein, standard method. It's very sensitive. So this is the ELISA, unfortunately, is a bulk method. So the ELISA results show that the ELISA cannot detect the early stage. You see the p-value is 0.11. So, and also this ROC curve to show that this use the fraction of the phenotype exosome, we can detect the breast cancer at early stage. However, the standard ELISA, they can only detect at stage 3, not stage 1 and 2. So I hope this very short introduction about our technology, and you have some rough idea. If you have any questions, you can always, email me, contact me. I will be very glad to answer any questions you may have.
All right, thank you, Dr. Wong. You have a whole bunch of questions coming through in the chat. So I'm going to go ahead and ask the first one, and then if you would like to go and answer the other questions, I think that would be great. But the first one, how specific is your antibody immobilized on the gold slide for CD81, for the CD81 marker? Do you know the level of affinity?
Yes, so we have done all kinds of specificity study. We're not shown here. So that's there are exosomes that are specific for exosome. We tried the IgG, they're not able to capture exosome. So the specificity of the functionless gold slides is exosome. So whatever the exosome, not a macrovesicle, not other. So they, as long as they have the CD81, so CD81 expression exosome.
Okay, great. Thank you. And if you have time and you don't mind, go through the chat because there's several questions. There's a lot of people who are interested.
Okay. So I'm still looking.
See if you can answer them.
Okay.
So I'm gonna go ahead and move on to the next presentation.
Sure, go ahead.
If that's okay. Okay.
Yes, I can answer them in the chat. Thank you.
All right, thank you so much. All right, next up, Dr. Jennings.
Okay. Thank you, and I do have a brief presentation, so
Yep.
if you make me focus.
Yep. Hang on 1 second. All right. Coming your way right now.
So, thank you for inviting me again to this program. I'm going to talk a little bit about this invention that we came up with when we were working with types of tissue scaffolds, particularly Kitazan sponges, for orthopedic applications and also for soft tissue applications. So here is an image of what some of these Kitazan sponges look like. And so, Kitazen is a renewable biopolymer. It is derived from a protein or a material, a polymer that's found in the shells of crustaceans like these shrimp and crab and lobsters, but also in other sources naturally like mushrooms and other types of fungi. And so Kitazen is derived from chitin, which is what is in these hard shells. And so once it is produced as Kitazen, then it has a lot of versatility for making types of materials, particularly types of natural materials, ones that we can use as implants. It also has the advantage that it's a renewable resource, so we're not synthesizing anything, and it also has a strong tendency toward the body recognizing it as something that is natural, and so the biocompatibility response is really great for Kitazen. So, we are interested in the field of biomedical engineering and tissue engineering and regenerative medicine. So that would be providing some support for tissue to regenerate and to heal naturally. So, one of the applications that we particularly have been interested in previously is the idea of bone tissue engineering, where if you have a gap or a fracture or a segmental defect in the bone, then at some point this is a little bit too much for your body to heal on its own. So just like scaffolding for a building, a tissue engineering scaffold, and in our case that is made out of Kitazen, can provide some support for that tissue to grow into and for that tissue to be built up and to provide some support for that healing tissue. However, once you have built your building and it's all nice and wonderful and it can support itself, then you kind of want to remove that scaffolding so that the aesthetics of the building and the actual function of the building can continue. And so that ends up being a major challenge for some of these materials like Kitazen, because they stick around for a long time in some forms. So, particularly when we've used things like neutralized sponges and beads. So, if that scaffold can't get out of the way once it's done doing its job, then, you might get some kind of interference with the healing. And so the healing can't take place. So because something is actually in the way. So what we wanted to design and to work on is a way that we could actually have a little bit more control over the degradation of these Kitazen scaffolds. So what the invention is just a simple post-fabrication treatment process that involves soaking these scaffolds in something like an acetate buffer or a controlled pH buffer for a specific amount of time. And so what that does is that it environment. And actually, we have shown and demonstrated that by using specific pHs of buffers, we can significantly increase the degradation of something like a Kitazan sponge. So the ones that we were using, this is a degradation study where we tracked the loss of mass over time. And so our regular scaffolds that we were using in the neutral condition were pretty much 100% there after 10 days. But by using some of these treatments, then we could stimulate that degradation so that the material gets out of the way and the tissue engineering application can then be enabled. So what is great about this technology is that it could be applicable for many different forms of scaffolds. So for skin tissue engineering, you're going to need something that's going to support soft tissue. For bone tissue engineering, you might require a little bit more strength. And so one of the advantages of Kitazen is that it is very versatile, and we can form it into something like this, very soft, spongy material. We can also make micro beads and nano beads from the material. We have used it in an injectable form, and very recently we've started to explore some applications of electrospun Kitazen membranes. Also another advantage is that in addition to the healing properties, we've also found that some of these forms are really great at delivering drugs and therapeutics such as growth factors, proteins, antibiotics, antimicrobials. So they have a lot of advantages if we could just get them out of the way. And to give you an idea of some of the applications that could be enabled by this technology, so this image is showing something that would be a bone tissue, a guided bone regeneration membrane that would be designed to maintain the barrier between bone and soft tissue. And that would be particularly applicable for things like dental applications where these guided bone regeneration membranes are used to manage the healing and facilitate the healing of bone graft. My lab have been exploring is the potential of these guided regeneration membranes to be used as wound dressings or skin substitutes. So, just in summary, our invention allows for a controlled degradation of Kitazen-based tissue scaffolds, and those have a lot of applications. So, it's a simple technique, it's a simple soak procedure, and we actually have shown that it does have some effect both in vitro and in vivo. So, here on the right, I'm showing some, one of these Kitazan sponges that has been implanted, and you can see that tissue is able to grow into that sponge because the material is actually going away. So we don't want to block any kind of healing. And so with that, I will open it up to questions. And I will stop my share so that I can see the screen.
All right. Thank you, Dr. Jennings. Let's see, are there any questions? What stage do you soak Kitazen in buffer?
So this would be after the material has formed. So, if you were using a sponge type of implant, then after you've neutralized the sponge, then you would soak it in the acetate buffer. And we found that specific amounts of times for the soak are particularly effective in controlling how that response happens. So it's kind of a post-fabrication process.
And is that the same if you electrospin it? You wouldn't include it in the solution, you would soak the membrane afterwards?
Yes. So, after you formed the membrane and then treated it either by neutralization or, we've been exploring some other types of stabilization methods, then you would soak it in the buffer.
All right, looks like there's one more quick question. Do you ever see any unexpected immune responses?
Well, we always expect some immune response, but our goal is to not have any unexpected or negative or kind of increased immune responses. And so we have done some studies in vivo for this, and I don't know the exact numbers, but these are not any larger than what you would have for, say, a neutral Kitazen material.
Okay. All right. Thank you so much. And with that, the next presenter is Dr. Min from St. Jude.
Yep, I have a slide.
Okay. Let me make you co-host. Hang on one second. All right, Dr. Min.
So can you guys see that?
Not yet. Now we're starting to see it. There we go. Development of novel and potent JK2.
Yep. Thanks. Alrighty. First of all, thanks to Chad that actually introducing this program, and thanks to Ryan for encouraging me to participate in this beautiful program. So today I'm going to share, first of all, it's my great pleasure to be part of this great program, and I'd like to share our IP actually generated from JAK2 degrader project for the treatment of COP2 rearranged ALL. I'm working at St. Jude, and I'm leading the chemical biology center here at CBT. And so based on what is the ALL and what's the burden of ALL, as we know, based on the reported database, actually out of the some of the causes of childhood cancer, there are top more than 30% leukemia, actually one of the major burden for ALL. And furthermore, actually the relapse, as you can see on the bottom. So let me just change the here. Relapse in ALL, actually one of the other main cause of the deaths for childhood cancer. And there are historically multiple drug actually has been developed for the ALL, as you can see on the right table. And recently, several kinases like Glioblastoma and Tarsanitinib, some of the kinase, and on the top of that, some mono antibody therapy and some CAR T therapy has been used as a frontline treatment. And but still, there is some drawback because Philadelphia-like ALL is one of the main devastating form of ALL. And because of the COP2 rearrangement and the overexpression in 10% of BRAL, and because the 5% of Philadelphia-like ALL has been found, and this is closely associated with the poor clinical outcome of previous, like a frontline drug. So that actually causing lacking efficacy in the therapy. And on the top of the genetic alteration in the COP2 rearranged cell line. There is also JAK1/2 kinase mutation has been found, and as a result of that, this causing JAK-STAT signaling pathway activation at the downstream. So overall, because of these genetic alteration and the mutation in this Philadelphia-like ALL, existing FDA-approved kinase drug like a JAK2 inhibitor actually shows very limited efficacy and lack of efficacy. So there is an unmet need for new therapy and new mode of action and the new chemical compound that can cure that. And so here I just wanted to quickly introduce the PROTEC technology, what it is. So this is basically this is very innovative and emerging technology recently developed as a new therapeutic modality, which is very fast growing now and highly competitive in this in both academic and industry area. And using this technology, the main difference is in between conventional drug and PROTEC is conventional drug is occupying the target of interest, protein of interest, and they inhibiting or activating and changing function based on occupation. But in terms of PROTEC, this is proteolysis targeting chimeras, actually conjugating two molecules, one for the target protein, the other for E3 ligase ligand, simply conjugate with the linker. And then what they're doing is they binding to the target, and then they hijacking E3 ligase in our system. And then E3 ligase, what they're doing is get along with the ubiquitinated source, and then transfer the ubiquitin group to the target of interest, and make a polyubiquitinated protein. This misfolded protein recognized by proteasome and then degraded. One of the fascinating science over here is different with the conventional drug candidate. This PROTEC compound actually can be reused, go to the catalytic cycle, and then go targeting the other same target molecule as well. So there's still people normally call it as a novel novel therapeutic modality. So our hypothesis is by adapting this PROTEC technology, we will be able to expect the total suppress the downstream JAK-STAT pathway signaling by degrading JAK2 kinase instead of just inhibiting. And as I explained, the rearranged ALS cell line normally they are stabilizing by inhibitor and hyperphosphorylating and activating JAK2 STAT pathway. But when we degrading the target from the proteasomal system, then we're going to totally expect to shut down this downstream. So we designed the PROTEC based on two FDA-approved compounds, ruxolitinib and baricitinib, which is JAK1/2 inhibitor. And to design PROTEC to incorporate E3 ligase on the JAK inhibitor through the linker, we need to investigate it, elaborate the position of linker. So we actually obtained the crystal structure from both ruxolitinib and baricitinib against JAK2, and we figured out the position of linker we're supposed to put it in. So over here, I'm just showing the representative protein we made, but from this project, we synthesized more than 50 different protein. So as a biological profiling, you can see the in vitro and in vivo activity. Surprisingly, you can see in terms of cellular activity, our protein compound showing like more than 5 order magnitude potent activity compared to the ruxolitinib, which is JAK2 inhibitor and FDA approved. And also we confirm the target degradation profile that showing JAK2 degradation time the dose-dependent manner from Western blot. And we also compound the in vivo efficacy using the COP2 xenograft model, using bioluminescence assay, and then human-dose inhibition. So as a summary, I think we actually were successfully developed a series of heterotype functional prototype utilizing simply two different FDA compounds. One is JAK inhibitor, the other is cerebral binder, IMID. And then we built a new molecule and showing totally different mode of action with different therapeutic modality. So we are expecting to obtain some improved clinical outcome in the near future for the successful treatment of Philadelphia-like ALL, which is, as I mentioned, one of the most problematic childhood and adult cancer in ALL. With that, I'd like to thank you for your attention, and I'll be happy to take any questions.
All right, thank you, Dr. Min. So first question here is, how specific is this PROTAC for JAK2? Are there any non-specific degradations?
That's a great question. That is always main concern in this field, and definitely we did TMT as a proteomics study. So we knew this compound, that some of the lead compound is very specific against the JAK isoform, the JAK2 out of the isoform. So we confirmed that.
Are there any other questions? I guess I could ask one. You might have briefly mentioned it, but how big and flexible are the linkers?
That is also a great question. The linkerology is one of the key features in this field. Like this kind of strategy of life antibody conjugation and the CAR-T therapy as well. It's the same here. Like a linker length and linker components is very critical to different targets. So there is no gold standard in terms of length, in terms of component. Every single target has a different optimized linker. And also it's been known there are 600 E3 ligase. And out of that, now people using most likely 4 to 5, maybe less than 4, E3 ligase. So it's a really try and error and optimizing, spend more time based on SAL study and the medicinal chemistry effort need to be done.
Okay, great, thank you, and be sure to keep checking back in the chat for additional questions. I think I see another one coming.
Got it. Thanks.
All right, last but not least, Dr. Ma from St. Jude. Dr. Ma, are you here? No response yet. So I know he was excited to share his work and be a part of the program, so if he's not here, something must have happened. So I can go ahead and link a description to his work, and I can briefly read the description about his technology that he's working on. I think that may be the best way to proceed. Let's see. All right, so I have the description in the chat box. And his technology is called sequence error. So here's a little bit about what they're doing. So precision medicine relies on precision diagnostics, mostly through high throughput DNA sequencing in oncology. And researchers at St. Jude have developed a computational method to enable more precise measurement of sequencing errors. So their study revealed novel insights on sequencer errors that can lead to improved instrumentation. And NGS chemistry and ultimately higher DNA sequencing fidelity, resulting in software which can efficiently first determine if an instrument is working properly, determine if a specific experiment is done successfully, remedy a problem if the problem is not widespread in a specific experiment, and also serve as a calibration index to better develop instruments. Let's see. So they have basically developed a novel computational algorithm to measure errors caused by sequencers. And so what they've done is looks like almost 3,800 publicly available data sets from 75 research institutes. And they discovered highly reproducible patterns of sequencer errors. And what they were able to do is, looks like they were able to show where these errors were and then remove outlier and error-prone tiles improved for sequencing accuracy. And so based on their observations, they were able to produce a general purpose algorithm, which they call sequence error, spelled sequence with 2 R's, to computationally suppress sequencer errors and to effectively monitor sequencer anomalies. Let's see. So I think that is about the best description I can give. And so with that, we'll go ahead and wrap up the IP parade. I want to thank again all the faculty mentors for presenting their work. I think we have a fantastic panel of technologies, and I think everyone,
Hey Ryan.
I think this is, what's that?
Dr. Ma just joined.
Oh, he did?
He's probably getting his audio hooked up right now, but I did just get a new request to come into the room from him.
Oh, perfect. Hopefully he didn't hear that.
I thought you did a pretty good job. I'm putting that number 1 on the one team I want to work with. Yes, he is online. Dr. Ma, can you hear us?
Yes.
Okay.
Let me turn off my... I'm so sorry. So sorry. I was doing a really programming and I totally forgot this meeting. I'm so sorry.
That's all right. So Ryan Hughes just went through your high level, but we'd like more detail from you. So do you need to present anything? Yes. I can make you a host.
Yes. Do you mind if I can share my screen so that I can...
No, don't mind. I'm making you a co-host right now. You should see the ability to share right now.
Yes. Thank you so much.
Yes.
And, I really apologize for my oversight. This is really... I'm so sorry.
No worries. We're all excited to hear what you have. Go ahead.
Yes. Okay. I'm going to be sharing my screen now. All right.
Yep,
so
we see it.
Can you see my screen?
Yep, cancer early detection.
Cool. Yes. Okay. Hopefully, I can deliver my what I want to talk quickly. So basically, the overview, the life is essentially a growth. So we start from single cell and then get more cells. But this procedure is not perfect. And sometimes the daughter cells may have mutations and due to either replication or repairing mistakes. So this is broadly called genetic heterogeneity. So this kind of phenomena is very, it's almost everywhere. So in biomanufacturing, so here you really want to make sure that your product has a high purity. So such as you want to generate antibiotics, or maybe even just producing beer, right? So it's also relevant to agriculture, such as drug-resistant pests, or for us, it's mostly medicine. So such as infectious disease, you probably already hear that the COVID variant is the COVID virus is mutating, right? And in our St. Jude Children's Hospital, it's mostly cancer stuff. So let's talk about cancer. So the typical disease procedure of a cancer is like a diagnosis relapse. But before diagnosis, there is already something, the cells is already growing. And after the diagnosis, usually we apply very strong therapeutic pressure so that the disease can go away for a little while. But sometimes there is a drug-resistant mutation coming, and then there's a relapse. So DNA sequencing is really the key ingredient for us to uncover this kind of heterogeneity. However, the accuracy of DNA sequencing is a big challenge. So one of the good analogy here is this image analysis. With a high level of noise, you cannot see anything, but when the noise level is decreased, you can see Charles Darwin very easily. Very similarly, in this DNA sequencing field, people think that there's a high error rate in the DNA sequencing. In the early 2011, it's really high, and in the whole past decade, it's still very high. Only in this 2019, we published our paper to push this error rate much lower. And let's look at it on the industry. So I don't know if you guys know this Grial company, which is a spin-off from Illumina. So you can see what they are trying to do is essentially early detection of cancer. So what people already know is that if the disease is diagnosed at a late stage, the survival rate is really low. But if you can detect the disease much earlier, you can get a much higher cure rate. That's a key. So the Grial already get a huge amount of funding after I think it's almost 4 years from its beginning. So let's look at the Grial technology. In this late 2019, they published this paper with MSK. And if you look at this Nature Medicine paper, many of the authors are actually coming from Grial. So essentially, I would say all the analytical stuff of this paper is representing the state of art of this Grial technology. But if you look at their data, basically, whenever you go to this 0.1%, the reproducibility is really poor. I would guess probably 0.2% is their limit of detection. So we apply our methodology on their data, and we can find almost everything they found. We missed 4 that they have, but we found 27 more that they can do. This data set is not ideal testing ground for our methodology, but you can see we are already doing better than them. So our methodology has been applied to multiple studies. Essentially everything is on cancer, so which is really cool. So after that 2019 work, in this 2021, we published this new methodology, which essentially is trying to calibrate, to look into how the instrument is working. So this one, I would say has an even broader impact to this field, because it's impacting how the service providers such as DNA sequencing company and the instrument manufacturer, including Illumina, and of course informatics service such as us, so who is analyzing data for the patient. So in conclusion, I think that we have really good technology, and we are still continuously developing these novel methods to tackle this challenge. Just as I was so late because I was just excited to having another really nice program in this afternoon. So basically, we are having more product coming out. So our goal is to understand the role of this genetic heterogeneity in cancers, and eventually this is our mission, because this mission makes sure that our lab, our whole development is very tightly related to make it really clinically applicable. So that's the overview of my technology.
All right, thank you, Dr. Ma. I mean, you made it just in time to present. So I think.
Thank you so much.
So we've been asking the audience to limit it to 2 questions. So they can either ask it in the chat, or they can ask the questions themselves. So if you don't hear any questions in the next minute or 2, just keep looking at the chat because they've been popping up throughout the evening.
Cool.
Nothing yet. I guess I can start with one. So do you see this as
sort of as like a software where people can send you their sequencing results, and then you can find these heterogeneities, or would you sell it to other companies to do it themselves?
Okay, that's a great question. So I would say this is not a one-shot field. So this field really need a much higher sensitivity than we currently can do. My lab's technology, I would say it's number one in the world so far, but I still say it's not satisfactory because according to our calculation, every 10 times error reduction equals roughly 1 month of early detection. So our current sensitivity is roughly around 10 to the minus 4, which probably is maybe 3 months something early detection. I really want to push it into at least a half year or 1 year. So that will be roughly 10 to the minus 6, which is what I'm doing right now. So of course, when this paper is published, we publish this software so people already can analyze their data using our methodology. But still, it depends on who is analyzing data, and that is still a pretty critical dimension. So what I envision is there need to be a company to provide such kind of expert service. Not everybody can do it excellently, right? So people can already use it. For example, this instrument, we found that some of the instrument, people think it's great, but we can find that there's a huge amount of problem of that instrument. If the machine is not working well, you just cannot get a great result, right? So basically, I would say people can use our software, but it still depends on who is analyzing data. So probably in the short term, in the first several years, suppose we have a startup, I really want to provide the service by ourselves. We want to know, we want to diagnose the problems, make sure that everything's working perfectly, and then if we have a super nice pipeline, people can take that pipeline away, and then they can do it by themselves. But at the beginning, several years, I don't think that is easy to achieve because there's still many problems people don't know yet.
Right. Okay, great. Thank you. So, again, keep checking the chat to see if there's more questions, but I think with that, we're going to go ahead and thank all of our faculty speakers. I think we have some fantastic technologies to work with, and also some fantastic mentors for the participants. So with that, I think we're going to ask, or maybe, Brian, do you want me to turn it over to you to explain?
Yeah, I'll take it over from here. I got it. Can everybody hear me? Okay, so here's the time when you get to submit your sessions. So have we, Shelby, have you sent the link again for the IP submissions? Okay, so Shelby just put it into the chat for anyone. That's a link that you haven't looked at previous to now. If you could go to that link and fill it out and leave which ones you want, or put which ones you want on there, 1 through 6. If you just want to do 3, that's fine too. And send that in. Shelby is going to take from now until the end of our sessions here to collate that information and get back to us with the teams. And everyone's more than welcome to stay, get some questions here from the IP presenters. You're more than welcome to stay and hear what happens from here this point out. But you'll certainly benefit from our work as things progress. So thank you again. I'll repeat what Ryan said. Thank you for presenting. And if we have any other further questions, we'll follow them on if you're not on the line and listen to those. Okay, so you have the next 5 minutes to take a break and do something for me while you're doing it. If you haven't submitted your IP request, I'd like you to do that first. And then take a small break, bio or otherwise, and find something in your house or wherever you're at that best describes you. Some sort of object or something. Please make it appropriate. We don't want anything, inappropriate, please. But just something that best describes you, and we'll hold them all up in our screens when we are ready. But I have 7:48, so we'll take until 7:53 to do that. and we'll come right back. So everybody take that 5-minute break, look for that, fill out that form, and we'll see you in a few minutes. Okay, everybody should be coming back. We got about 1 more minute. Okay, everybody should be coming back. We have about 1 more minute. And I already said that once, but I was on mute. So, like I said, that's the words I've said the most this whole year.
Hey everyone, responses are coming in a little slower than anticipated. So, of course, take your time to make sure that you are choosing something that you want. But get those in as quick as you can too, because it's first come, first served. So I don't want you to not have a preferred IP to work on because it took a little longer. So the faster you get them in, the better.
Okay, we're going to try this. I would like everyone to turn on their video. We'll try not to break Zoom with our 30 plus people. Oh, I see one live animal. I was wondering how many we're going to see. If I could go find my cat, I would do the same. All right, so on the count of 3, we're all going to put in front of the screen the thing that best describes our personality. Oh, wait, I'm going to switch to... Hang on one second. Okay. Ready? 1, 2, 3. Coffee mugs, see those? I see you. Am I the only Lego? I see a cute, pretty little girl here, and a cute dog as well. The girl is much prettier though, but I do love the hound there. All right.
Brian, fun fact, that's my daughter.
Oh, that is okay. Very good on you there for your wife and your daughter there. Yeah, so I have a little bit of a Lego thing. So this is what I've been working on lately. Don't judge, but it's what I do. All right, thanks everybody. I think that was kind of fun and cute, and it worked out well on the Zoom. So if everybody wants to go back off their video and be able to relax, they can do that too and just listen in, or you can leave your video on. Your choice. I'm going to switch back to this mode and pin my video. Okay, well, I don't think we're quite ready for the team reveal, so we're going to come back to that. Shelby is working on that. Please, 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 as we progress through this, we don't have much more time. We're already about 5 minutes to when we were supposed to end, but we are going to work through the last little bit of this. But please, please get your team selections in as soon as possible. And if you don't care to choose and you liked all the intellectual property, then you don't have to submit a form, and we'll just put you in the same spot, in the spots. But if that's the case, still go ahead and put up in the chat to Shelby that you don't care what team, and she can just place you wherever she chooses. And we'll come back to the team reveal. Okay, we're going to try an icebreaker number 3, and this is going to involve the form that we did earlier, and I'm going to pull that up right now. All right, so we asked everybody to submit a word to describe, how they feel about the intellectual properties. We did that before the intellectual properties, but just what you were looking for in the work that you're going to be doing. These word clouds are kind of fun, especially when there's a lot of duplicates and you see the words get bigger. But we do have some pretty cool words here, so everybody submitted some unique stuff. I'm assuming I can define some of these things, but change agents, disruption, value to your customer, security, and your technology, sustainability. That has a great a bit to do with what many companies are working for these days of trying to introduce sustainability into their operations, zero footprint, so on and so forth. Hope, I love that one. New things, therapeutics. I mean, we've talked about vaccines all the time. Therapeutics are also very important for those that are not able to get the vaccines and everything else, just for general care. Solutions, comfort, and innovation. One of my favorite words there at the end of just being innovative and building innovations. And that's really what entrepreneurs do, and that's really what scientists do, and that's really what a lot of you on the Zoom session are doing as well, that are going to take part in this cohort, is to be innovative. Our groups that we represent, the entities, the institutions that we represent, are in the business of creating new technologies. UT, St. Jude, University of Memphis, no exceptions, been doing it for decades. The countless examples of innovations that we could talk about for St. Jude and all these other entities is just amazing. So we want to see this intellectual property that we have going forth right now in the same vein. I thought, being not a scientific person, but being a technical person, I found a lot of great things. I love the sequencer error thing, especially the fact that it's not just in life sciences, and you illustrated a great example of beer, which is also one of my favorites. So, anything that has general sequencing and testing and looking for errors, that's only going to increase, efficiency, efficacy, everything you can with a solution. So that I would put that as number one. I can't work on any individual team, Dr. Moss, but that's certainly the one I would work on as number one. I like the other ones as well. Okay. So let's go back. All right, so there's a few things I wanted to discuss as we move towards the tail end of this session, just really how the weeks are going to progress. You can look at the schedule that's up on the Scipreneur Challenge, Memphis Scipreneur Challenge website. It has all 9 weeks there. We're going to step through a lot of content in the next 3 weeks. It's really going to be setting the stage for all the components of information that you're going to need to build your narrative, to understand the problem that you're trying to solve and the solution that you're going to apply to that problem, the customer segmentations that we're going to focus with your solution, because everyone's segmentation could be different. They all could be similar, but generally they're going to change based on what value propositions your customer targets are going to benefit from by utilizing your product or service, whether that's a therapeutic or some sort of error sequencer solution that we just talked about. So, that's where we really are going to be working on, is understanding how to build that narrative and understanding what we need to do as entrepreneurs. Obviously, we're not going to be able to just completely spawn new companies in just 7 weeks, but we do encourage y'all to work to that intent. And what happens after this session, after the final presentation, who knows? And the support and resources that you have at your disposal do not end at the end of this session. Shelby and myself, Ryan, Erica, these are things we do year round for Biotin and the other areas that we work for. So this is what we do. So we'll certainly be resources for the long haul with y'all as you progress through these ideas and launching businesses potentially. So one other thing I wanted to discuss was just kind of general housekeeping. This is kind of the format that we're going to be going at week to week on Zoom. We'll be making some tweaks and modifications along the way. If you have some suggestions, I'm all ears on how to make this experience the best that it possibly can be. We do not, prescribe, for lack of a better term, any certain tools that you need to utilize to interact with your team. I highly recommend, Zoom and WebEx or any other kind of web conferencing solution. And I also recommend y'all doing weekly, if not 2 a week meetings through this process as a team, because there is going to be a lot of work that you're going to do offline to be prepared for what we discussed the previous week and then what we will discuss in subsequent weeks. So WebEx, Zoom, free conference call, whatever you want to look at. As far as communication goes, obviously there's email, text, and phone. Discord, you can generate a Discord server. Slack, if that's something that y'all use as well to communicate and to collect your information. Google Docs, Sheets, and Forms and Slides are great tools to be utilized by teams. You can work on the documents at the same time and track all your changes and to store those in a repository that everyone has access to, including those that, from the facilitation team that if you want to bring in, we'll be able to access those documents and give you advice as well. So you as a team will need to figure out your best mode of operation to work through this session. And we can help make suggestions, but we're not going to select anything for you because everyone has their personal preference. With that, for the next 5 or 6 minutes, I'd like to open it up to any questions that you have about the program. Let's stay away from questions around the specific intellectual property because we're going to get deeper into that, and you can submit those to the individual IP owners. But if you have any questions for Ryan, Erica, Shelby, or myself or about the process, let's discuss those now. And remember to take yourself off mute.
How much is the prize money again? I just want to reiterate that. Find out how much we're talking here.
$1 billion. No, I'm just joking.
Yeah. So for first place, each member of the team will receive $500. For second place, each member of the team will receive $400. And third place, each member of the team will receive $300.
but every participant will receive some sort of monetary prize. I think we've given out $25 to $50 gift cards for everyone in the program. We're in the process of designing some really cool swag with like some coffee mugs, T-shirts, things like that. So anyone who participates, you're not going home empty-handed.
Awesome.
Y'all, this is Chad from St. Jude. I was going to say that in the past we've had a lot of support for this, and the prize money could have been made bigger, but it creates problems for international students who are here on student visas. So that's why it ends up being spread around and at the $500 level.
That's a lot of money to me, Chad.
Hey Chad, good that you pointed that out. I just want to make sure, because we ran into it, so just want to make sure that everyone here are aware. It could be $499, not $500. So just make sure you guys know about that for international people. Anything less than 500. So we had to keep it at $499 for international people. So just.
Yeah, that's true. Sorry to mislead you, Brad. It is $499.
I know this because when we, the checks for 500, they had to come back and we had to reissue things. So
redonate that penny back to Ryan.
Yeah.
Other questions? Okay, as of the last check, we had 11 people submit the forms. We have 25 people online. We should still have 4 or 5 more participants. So if you did not fill out a form and do not care what team you're on, go ahead and submit a chat to Shelby and let her know that you just didn't fill out a form, but you don't care what team you're put on. Other questions?
I have a question. Ryan, back when you went through Scipreneur, your first round, or first time through, what was your project on, and maybe talk about your experience back then and the value you got out of it?
Yeah, that's a good question. So I first went through the program 2 years ago when I was still working at St. Jude, and our technology was working on CAR T-cell therapy for treatment of cancer. And really, I think it was the experience that I had with Scipreneur, with the relationships that I built, the mentors that I met, that really pushed me to sort of step out of my position as a postdoc at St. Jude and join a program at the University of Memphis, which is really focused on early career scientists who want to start companies based around intellectual property at the university. And so when I started that journey, and ultimately when I started my company, the first people that I reached out to, or the first people that reached out to me to congratulate me, were the same people who helped me in Scipreneur. So, it's really
product. Is that it?
Yeah, patents to products. So, I mean, for me, and I think it could be the same for a lot of people, it's sort of a launching point for individuals who don't realize that they want to be entrepreneurs until they go through this program.
Can you give us a little rundown about you, Erica, and who else is in that program, if you want to describe them briefly?
Sure. So just briefly, the Patents to Products program is an initiative at the University of Memphis that I briefly described, but we really recruited really diverse in backgrounds of people who wanted to start companies. So my background's in biochemistry and structural biology, but now I'm working on technology in biomedical engineering for wound healing and inflammatory diseases. Whereas you have Erica, who's now the president of BioTN. And Erica, are you still on here? I don't want to steal your thunder. If you want to talk about what you do.
Sorry, my computer was on mute, and I, for some reason, was pressing the button frantically. But yes, so just you're talking about the Patents and Products program. My background is in medicine and research as well. I'm basically considered a physician scientist. I did an MD PhD at UT. And so my role in the program is really focused on healthcare technology, postoperative care, specifically in monitoring, remote monitoring technology. And so I am developing some technology that will help predict whether or not a patient will develop blood clots after they've had a procedure done. So this has been literally like my first time ever jumping into, anything business related, anything industry related. I mean, with medicine, you train for such a long time and very, very focused on clinical science. And so this has been actually, I wish I had this experience before, like Ryan did when he was going through the Memphis Scipreneur program. But I've really been learning a lot from him just because of his experience with Memphis Scipreneur, and it's really been helpful. So I think this is an opportunity for you guys to really, really jump in there and learn how to do this, and maybe even utilize your experience here to do something, a little bit more on a larger scale, like a biodesign accelerator program or something like P2P. Definitely has been a wonderful experience for me. And I'd be happy to, like, if anyone's here who is, kind of more interested in just learning about that experience and kind of how I've veered into something a little bit different outside of clinical science, definitely reach out to me. I'll drop my email in the chat too. Be more than happy to talk to you.
Yeah, same here.
At the UM, I guess called the Research Park, I think I saw a couple patent and product desks up there, like off to the right when I venture up there. Is that where you guys are located, Ryan?
Yeah, you definitely did see a couple of our cubicles.
That's where Sweet Bio currently is located, right? So do you know much about the Research Park, and I guess what connections they have to U of M and the background from their side?
I mean, it's probably easier to talk about that offline.
Yeah.
I can tell you a little bit more about Research Park. It is affiliated with the university, and they're really focused on recruiting, local startups in the Memphis community to try to grow that ecosystem. And they give them, really, a nice community of entrepreneurs for low cost and, really not a lot of money to be there. So I think it's a great atmosphere. You're constantly being, interacting with different entrepreneurs. So I think it's a really good initiative that the university did.
If you want an outsider's take on it, who really, really likes the University of Memphis, but I think the short of it is they've got a guy named Jasbeer over there that is really, really, really working hard to check off boxes, because Memphis is just outside of the long list of schools that are tier 1 research institutions, and they're so close. Like, he's sort of charged with getting them there. So there's a lot of activity to get us, we'll be low on list, but, getting over the hump and on the list and then moving up. I think there's really, really, from leadership on down, a real charge to make University of Memphis a tier 1 research institution and have it mean something.
And Shelby, I see your question about 11 signed up, so I will, go ahead and put this out there for all those who did sign up. If you know anyone else, at your work or, in your lab or whatever it is you do that would like to participate but couldn't make it this evening, let them know, because we're more than willing to, accommodate them and allow them to jump in before next week. But as long as everyone can kind of get their teams formed by next week, we can do that. We can make exceptions for people who couldn't make it today.
Yeah, so, Ryan, what you just said, Erica, Shelby, and I, we've all been talking offline. So what we'd like to do is just exactly what Ryan laid out is we're not going to reveal the teams tonight. We're going to give a little bit more time for people to submit the form if they so choose to do so. But what Ryan just said, a little bit about a word of mouth going out there. I mean, you've seen what we're all about and what we're going to offer. It's a unique offering. Not every city, not every region does things like this. I think it's great how we have 3 major research institutions here in town that are putting forth this much opportunity. And I think that's a big word and a very important word to utilize. And I think that opportunity is there with you as well to see how it, what it takes to be a part of some of these teams and these technologies, but really talk about how to best advise those institutions on how to launch these products. Too many products like this and technologies get left on the table because no one knows how to launch their business, or the institution itself is not in the business of launching businesses. They're in the business of, with St. Jude, again, curing, life-threatening diseases, terminal diseases, and infectious disease for children. That's their business. So launching technology out of institutions like that is often difficult. Even University of Memphis, which has a large research as well. So this is our opportunity to show these institutions that we can take that technology and we can bring it to the market. Is it going to happen in 9 weeks? No. Will it happen with some of you? Hopefully, if we stick to it and do this. So this is a real opportunity. So if you know what it's about, you know what we're going to do. If you feel like this is an interesting thing that you would like to share with other individuals, like Ryan said, I think that we all think that'd be fantastic. We're going to give a couple more days, at least tomorrow, to kind of collect more names. We'll present the teams as soon as possible. It might be early next week. It might be the end of the day tomorrow. But like I said, we want to get as many participants as we possibly can to be on these teams, because it's difficult to do these teams when there's only 2 or so individuals on the teams. But we will make do if that's the case. So anyway, pass the word around, bring people in. I'm going to give you some links to some contact information here just right now, actually.
Right, we understand, with the week that we've had,
it's crazy.
A lot of people don't check their work emails when they're not technically at work, so we're hoping that, more people will be responding in the next couple of days.
Okay, so
people.
Go ahead.
Go ahead, Brian.
No, it's all right, Brad.
I was just going to say I might have a few people in mind who might want to do this. I might take it offline or talk to you with Brian.
Yeah, definitely.
All right.
Okay, so our main email address, and correct me if I'm wrong, Erica and Ryan, I just pulled this off of what I thought we were using, but is this the right email address for everybody? Memphis Challenge or memscichallenge@gmail.com?
The mem site, I thought we changed that, Erica. I thought we need the biotin one.
Okay. We'll send out, you have...
For Scipreneur, we're using the Memphis Scipreneur.
Okay. Ignore this one, although just ignore it. And we'll send out the right one in chat. If Eric, if you could drop that in chat for me, that'd be great. That way everyone has it. But these links I do know are good. So the Memphis Challenge link, the Scipreneur Challenge link is right here, memscichallenge.org. Take a screenshot, write these down. We'll send them out as well. And then the Tennessee Academic Alliance is here as well. And then the Center for Entrepreneurship. This is a small link because our URL is quite massive, and we'll send that out. Shelby, if you can send that out in chat, that'd be great. And just so you know, Shelby is one of my dearest friends and colleagues, and she and my wife are also really great friends. And I've called Shelby Chelsea, which is my wife's name, 3 times tonight, if you're counting, which I do quite often to both of them. They're both gingers, so it's just to happen. So if I call Shelby Chelsea, you'll know what that means. And she responds to both, so that's great. Both of them do that for me because I'm older and senile. So anyway, that's going to be wrapping it up for the night. I can't stress enough how much we appreciate all of y'all for joining us, how much we appreciate all the intellectual property holders for joining us and presenting to us tonight. It was incredibly interesting stuff. I'm going to wrap up my end and hand it back to Erica and Ryan if there's anything they want to close out with, but I really appreciate it, and I look forward to working with you all through these next 9 weeks.
Yeah, I don't really have anything else to add other than, just to reiterate that we're super excited to work with everyone, and, we're hoping for, a great 9-week program. So get the word out.
Thanks. Yeah.
Okay, and we'll be sending all this contact stuff out as well. So just keep an ear out for us. And if you have anybody you want to pass them along to those emails that we just put in chat. And I'll leave it open. I'm going to leave Zoom open for a few more minutes just so everybody can grab that information. And other than that, if you have any questions, just send them to us directly. All right. Thanks, everybody.
Thank you.
Guys. It's been fun.
Bye-bye.
Brian, are you still there?
Yes, but I was on mute.
Hey, so Rohan just messaged me. He's one of the members in the PDP program, and he wants to participate. He was just asking me if there's any conflict of interest being a part of patents or products and also going through this program. And I really don't think there is, so I'm probably going to let him know that he's more than welcome to join.
Yeah, I don't think there's any reason to not as well. I mean, the more the merrier. We talked about that yesterday too. So if it's good for you and Eric, it's good for us.
Yeah, I mean, he's a member of U of M, right?
Yeah,
so
yeah.
Yeah, he's a fellow, so he
Yeah.
gets the requirement.
I just got a chat from Jason Moore. He's part of, well, he sent it to me directly. He wants to know if he can observe and participate. He's got limited time because he's working on a R25 grant and also teaching in March. So do either one of y'all know Jason? Okay,
I was
Jason.
going to say, Jason and I talked already, and I slotted you in for the team that you wanted, Jason. In my opinion, Brian, Ryan, Erica, feel free to chime in. In my experience, I think that you should be fine as long as it's communicated clearly. You set the expectation with the team that you're assigned to, and that we have other members on that team to cover you as well when you're not able to be there. I think that should be fine, though. I think we should know more, hopefully, tomorrow when the sign-up sheet is more hammered out.
Yeah, I just want to be totally frank with everyone because, there's, with all the things going on, I want to participate. I wasn't totally aware of what this was, and that's one of the reasons I came tonight, but I think it's going to be really useful because I'm actually our department chair of our research entrepreneurship committee. And
Great.
so I just thought it'd be really helpful. But my time is limited, and I just wanted to be totally upfront about that. And I don't want to put any of the teams at a disadvantage from the very beginning.
Yeah.
So
we appreciate that.
Just to observe, or
I think you just I think we'll just play it by ear, and you set the expectation of the time that you have available. And if it's not enough, you'll admit to that and tell them there's not much more you can do. And I think anything you can do will help, though. And as a side note, I mean, just because the position you just said and your contribution to UT and the entrepreneurship space, offline, Shelby and I'd love to talk to you about how we can support you and your efforts and everything else. So that's another opportunity for us as well.
that'd be fantastic. I would love that.
Yeah. We, just as a side note, President Shannon, our president, and University of Memphis, Mike Hoffmeyer over the Career Center for Entrepreneurship is someone I worked for and worked with, and Shelby and I have known for several years, and we collaborate on a lot of things. But President Shannon at CBU, and we've been very successful at that. It's almost like the Career Center for Entrepreneurship has 25,000 students to deal with. We have 1,000, and if you add up all the other universities and all the other institutions, it might make up the 25,000. So we feel like we have an opportunity to help out in the community a little bit more, and work with other institutions like yourself. We're working with Rhodes and Lamont Owen, and St. Jude and stuff like that, we'd love an opportunity to have the Center for Entrepreneurship, support you as well.
That'd be wonderful.
Yeah.
Because I know it was probably last year sometime about this time, they had some representatives from, I think it was the biology department at CBU, come over to our program and talk about ways to collaborate. I think this is another perfect way.
We just started our push to be a research facility and institution last week. So we are going, are we going to be R1? Never. But are we going to do a lot of research and work and be maybe a sub institution for other major R1 institutions? Yeah, that's what our goal is. So our life sciences, our health sciences, our computer sciences, and our engineering, for the most part, are all working on opportunities to build out research and build ourselves as a research foundation as well. Yeah, CBU's really going to change a lot in the next 10 years. Brad, you have a question?
Yeah. So whichever team Shelby put me on, how many people do I need to recruit and lasso?
At least one would be good.
How many are on there? 2? Yeah, 3. Here's how I look at. Here's how I look at founding teams. Founding teams, a single entrepreneur, which I've been guilty of several times in 30 years, is never going to be as successful as 2 people on a founding team. But the magic number that I have found is 3. So 3 founders is usually the right way of doing it. 4 is also good. 5 gets to be a little too many cooks in the kitchen, not for a program like this, but for an actual business. Yeah, it's less about equity and more about too many chiefs and not enough Indians in that regard. So when you have 3, you're able to usually find someone who's the tech person, someone who's the business and sales person, and someone who just likes to control everything, and that's your COO or CFO, right? And so that's how it works. So 3 is good. That's our magic number. 4 is great. 5 is also workable. In a program like this, the numbers don't matter as much. But
yeah. So you tasked me with
if you could get 1 to 2 more people, you'd be good to go.
Yeah.
1 to 2.
Yeah, based on how many people you have so far. 1.
Okay, thanks, Shelby.
Okay.
There's 11, right, Shelby?
Yes.
So we have 2 on every team except for 1 team?
We have 1 team that has no one on it now.
Okay.
Every team has at least 2 people on it, and then there's 1 team that has no one on it.
Which team is that?
It is the method for detecting circulating tumor cells and extracellular vesicles. I said that like I know what any of that means at all.
Yeah.
It's funny because that's the one that I was like, I feel like there's a couple of ways in which I'd like to use this technology.
Yeah, I think I accidentally said something correctly.
No, seriously, add me on that one for just to jump in the room every now and then.
Yeah, she got a ton of questions too, but I think a lot of them were other faculty presenters.
Probably.
Yeah, hold on. So can we recruit Ryan, or what are the limits here?
Look.
We're definitely going to be mentor.
Yeah.
I'm giving up my time to be mentors for a team, definitely.
Yeah, I think what Ryan just said is what we'll all augment all the teams to make sure that everyone is able to progress along the same lines. It'd be hard to stop Shelby and I from jumping in to helping out y'all individually and as we progress. It's just what we do. So it doesn't matter to me if we only had 11. If we have 15, that's great. If we have 20, that's fantastic. If we had 5, 6 teams with 2 to 3 people on each team, we're gonna have a great time.
Yeah, and again, I think this week there was a lot going on. You even saw one of the presenters forget last minute.
Yeah.
And he had been emailing me for weeks, super excited about it. So
I can tell you, I was a hell of a lot less busy before COVID than I was now. I find I put more stuff in my day, and I'm just like Dr. Moss sometimes, and realize at 7 o'clock I was supposed to be on the phone with Shelby. So it happens. It happens.
I had to jump off
Okay.
7:40 to take a work call.
Yeah. All right. I think that's about it. Anything else?
Yeah, well, I mean, everyone else can go. I just wanted to ask Shelby a specific question about the people who did sign up. Was there, who were those science participants represented from? Is it more St. Jude, UT? Are there any U of M students? Like, who do we need to really target? Which institution, recruiting wise?
You look and see. Let me check their email addresses really quick.
Yeah, I could probably recruit some U of M targeted people, Ryan.
U of M is the most difficult place to get emails circulated around because there's no direct way to do it. And there's a lot of red flags and hoops you have to go through.
The only thing is, like, they're probably not going to be current students if you're looking for current students. I might be able to, like, reach out to some of the professors that I had and, like, then reach their students that are in their classes currently and see if there's any interest, but what's your thought?
As long as they're graduate students or above, like undergrad, yeah.
So grad students and above?
So some of them it's hard to tell because a couple of them use Gmail accounts, so I can't tell what institution they're from. But it looks like St. Jude and UTHSC are the predominant participants that have signed up. We've got one Memphis.edu email address, and then the rest, like I said, I can't tell because they're Gmail.
Is the Memphis one Rohan?
No. He signed up using his Gmail, actually. Rohan did.
Okay.